Hello friends, in this video lecture I will be talking about the termination of protein synthesis. Must watch my previous video about the initiation and elongation process of polypeptide synthesis for an easy understanding. So termination occurs when the ribosome reaches a termination codon so that no tRNA will come and attach to the A site of the largest subunit of ribosome, and this process, the termination process, is carried out. So let me draw the structure. So this is the larger ribosomal subunit and this is the smaller ribosomal subunit, and so this is the P site and this is the peptidyl tRNA, and this the stop codon, that is the A site, and this polypeptide, the polypeptide chain is now joined to this peptidyl tRNA. So large number of amino acids forming this polypeptide is joined to this peptidyl tRNA by an ester bond, right? So this is the A site of the larger ribosomal subunit, and it has a termination codon. Let the termination codon be UAA, right? And in this stage, no tRNA will come and attach to the A site of the largest ribosomal subunit, because no tRNA anticodon is complementary to this stop codon. So the process of termination occurs here. So let's have a look on the component those are required for the termination process. So the following components are required. So first is the process requires the termination codon. Termination codon. So there are three termination codon: UAA, UAG, and UGA. They are also named as ochre, amber, and opal. So UAA, UAG, or UGA. Any one of these termination codon at the A site can bring about the termination process. So second, the major factor that causes the termination are the release factor. Release factor. So these release factors, in prokaryote there are three release factors, that is RF1, RF2, and RF3. And in eukaryote there is ERF1. So this is the sole class 1 release factor in eukaryote. And the third thing, that is the GTP, that provides energy for terminations. So these are the components required for the termination process. So let's see how this event occurs. Now the mRNA, this is the mRNA and this is the ribosome, and the A site has this termination codon. So this release factor has a major role in terminating the process. So let's have a discussion on this release factor, how they bring about the termination process. So let me erase it. So in prokaryotes there are three release factors, that is RF1, RF2, and RF3. And these three release factors are grouped into two classes. So first is the class 1 release factor. Class 1 release factor that comprise RF1 and RF2. And the class 2 release factor comprises only RF3, right? So what is the role of this class 1 release factor and class 2 release factor? The role of this RF1 and RF2 is to recognize the stop codon, and they also terminate, means also triggers hydrolysis of this ester bond. You know the ester bond that is formed between the carboxyl group of this last amino acids. So this one is the last amino acid. This is the last amino acid. So I have made this structure outside, this polypeptide chain outside, only for an easy understanding. But this polypeptide chain is remain inside the larger ribosomal subunit, right? So this is the last amino acid, and this is the tRNA molecule, 3' end, and it is the last nucleotide, that is adenine nucleotide, and this ester bond is formed. So let me draw it. So this is the ester bond. This is the ester bond, and the ester bond is formed between the carboxylic group of the last amino acid and the 3' OH end of the last nucleotide, that is adenine nucleotide. And this RF1 and RF2, they triggers the hydrolysis of this ester bond, so that the polypeptide is released. And the class 2 release factor, the role of this class 2 release factor, that is RF3, is to enhances the activity of class 1 release factor. And also this RF3 helps to release the RF1 and RF2 from the larger ribosomal subunit after the polypeptide chain is released. So they helps in dissociation of RF1 and RF2 from the ribosome. So this is the role of RF3, and it is a GTP binding protein. GTP binding protein. Right? So this RF1 and RF2, the role is to recognize the stop codon. RF1 recognizes the stop codon UAA and UAG, and RF2 recognizes UAA and UGA, right? So these are the role of class 1 and class 2 release factors. So this termination codons gives a signal of this termination process. So how this event occurs? When the ribosome reaches the termination codon, then this RF1 and RF2 come and attach to this A site. And we should know that the structure of RF1 and RF2, and in eukaryote ERF1, their structure, these are the protein factors, their structure is similar to the tRNA molecule. So they mimics the tRNA and come to attach to the A site, and they cause the hydrolysis of this bond. So if this is the polypeptide chain, this is the last amino acid, and this last amino acid has a N-terminus N, means the amino group. This is the P amino group, and this is the N-terminus N. And this one, after the hydrolysis of the ester bond, it becomes the carboxylic end, carboxylic acid group end, and this last amino acids have a carboxylic end, that is called the C-terminus end. And this reaction, these hydrolysis reactions, require one molecule of water, and the reaction takes place in the peptidyl transferase center, that is PTC. The reaction is carried out in the peptidyl transferase center, and the polypeptide chain is now released. So after the release of this polypeptide chain, RF3 helps in dissociation of RF1 and RF2 from the larger subunit of ribosome, and the two subunit of the ribosome get dissociated, and the mRNA undergoes degradation. So these are the events of this termination process, and this newly polypeptide chain is in the primary structure. So in order to achieve the biological active form, this newly polypeptide chain, the newly polypeptide must undergoes folding and processing with the help of special proteins called molecular chaperones. So in my next video, I'll talk about the post-translational modification of proteins. Thanks.